I'm going to tell you about the Norwegian multi-center double-blind and placebo-controlled phase 3 study using the monoclonal antibody Rituximab for induction and maintenance treatment in ME. We call it Rituximab in popular, and you see the trial code in clinical trials underneath. This is a joint effort between 5 hospitals in Norway. It is initiated and led by us in Bergen. In addition, Oslo University Hospital, Notodden Hospital, St. Olav University Hospital in Trondheim, and the University Hospital of Northern Norway in Tromsø are participating. And we have a lot of sponsors, which we will come back to later. Now, the background for this study is the information we've accumulated during the years. As many of you have heard of, we had a patient that had ME since 1997. In 2004, she was given 4 different chemotherapy regimens for lymphoma. And during one of those 4 regimens, she had about 7 weeks into the treatment, gradual or quite quick improvement, astonishing improvement of her ME symptoms. And this lasted about 5 months. And when she was finished her lymphoma treatment and the chemotherapy, the symptoms gradually came back. She was observed about 3 years, and then Dr. Fluge, who was her consultant at the time, took up the question, what had happened to this patient during this period? And we decided we would try to find out about it. And by looking at the drugs that she had been given, those she had response to and those not, we found out probably 2 drugs were responsible. It was methotrexate and ifosfamide. And looking at the drugs, how they worked, we found out they maybe had a relative B lymphocyte depletion as a mode of action. And we knew that that could be done in a special way. Then we knew what we did. So we decided to make it a small trial in this patient, giving her a single infusion of rituximab. She was more than willing because her ME was again giving her invalidity. We gave her the drug, nothing happened for about 7 weeks. Then she calls Dr. Fluge and says, I think something's happening. Calls back 2 days afterwards, something is definitively happening with me. And she had a response which lasted 4 to 5 months, and then the symptoms came back just after the chemotherapy courses. We, in the meantime, had talked to the ethical committee and were allowed to treat 2 other patients, also with rituximab, because rituximab, we didn't know if this was something very special with this patient. So both those patients also had response to treatment. And then we were at a decision point, what to do. Treating ME is not a natural thing to do in an oncology department. We had no budget for it. And doing a phase 2 study and then later a phase 3 study, we didn't have the time for that. So we decided to take a shortcut. We went directly to a double-blind randomized small study and speculated that the response rate would be rather high. If not, it was nothing to be bothered with. So we decided for 30 patients. It was quite a risky business. Of course, we did a shortcut, and we didn't know that the 2 first patients we had, they had an unusual early response to rituximab. They had response before around 2 months, which is quite unusual. Most patients, they respond after a lag time of 4 to 5 months before they get the clinical response. So the primary endpoint that we chose proved to be wrong. So even though 10 of the 15 patients that were given rituximab had response and only 2 of the placebo patients, the primary endpoint was negative. So in fact, it was a negative study defined from the primary endpoint. On the other hand, looking at what happened to the patients, everyone could see, and statistically we could also prove it, that the courses in these patients was different. So this was the first study ever on rituximab in ME, so it had its weaknesses. So we had decided when we did this study that the patients in the placebo arm that proved to be in the placebo arm had to be given an option afterwards to get Rituximab, trying to learn something more if the study proved to be positive. And that is why we went on to the phase 2 study, which is to be published in PLOS ONE in maybe 2 or 3 weeks. It's accepted. Some of you know the results from this study because we've, even at this meeting, talked on the results, but we had to have 3 years observation time before we could publish. So that's why it's taken so much time. The patients are very waiting. They're waiting very much for the results, and now they will be there. So it seems to us in this phase 2 study that manipulation of the immune system seems to result in clinical responses. But remember then that in a phase 2 study, you treat all the patients in the same way. We had no placebo arm here. So that's a weakness in that study. But we saw the same thing as in the double-blind controlled study, placebo-controlled study, that 2 of 3 patients, 64% of the patients, had clinical responses according to predefined criteria. And we also saw that the duration of the responses were much longer when we gave maintenance treatment, which was the essential point in this phase 2 study. Not only induction, giving the drug at 0 and 14 days, but up to 15 months rituximab was given. Although this was not, there was no placebo arm in this study, 9 of the 15 patients in the placebo arm of the first study participated in this phase 2 study. And 6 of the 9 had clinical responses within the first year, which was the observation time in the first study. And the 7th patient also reached after 1 year a response. So 7 of 9 of the placebo patients in the first study, in fact, responded when they were given rituximab. We also saw that the quality of life in the patient that responded was very good in those that had major responses, and that was the majority of the responders. We saw a marked improvement in physical function in very many patients. In fact, some of the patients were completely back to 100% physical function. Although there were also other patients that maybe went from 5% initially up to 30%. And for them, that was really something that was important. Sometimes these patients are much more grateful than those that go from 20 to 100%. However, we observed something we didn't see in the first study. That is that when we gave the infusions, about 20% of the patients had transient worsening of their symptoms. In fact, single patients could have worsening of the ME symptoms while the infusion was given, which was, from a mechanistic point of view, quite interesting to observe. We hadn't seen that before. Although this was not part of the study, while we were doing this phase 2 study, we also explored methods for more objective analysis of patient levels of function. And we tested them in these same patients to get experience for future studies. For example, standardization of bicycle ramp exertion with ergospirometry and electronic physical performance registrations for 7 consecutive days. We also got very interested in something called endothelial dysfunction. That is that the innermost layer of the vessels do not seem to be functioning properly in ME patients. That is, they do not manage to give the signal to the muscle cells outside to relax as they should when given adequate stimuli. So this is an area that we also try to get some expertise in while waiting for the next study. Because it's been clear for quite a long time that we had to go on with a new study. When we learned more, we've known for at least 2 years that the response rates would be good, and we couldn't stop there. But of course, the world will not accept a phase 2 study, will not accept rituximab as a standard kind of treatment for ME based on a small phase, a randomized phase 2 study and a phase 2 study just giving rituximab. So we knew at the time that we had to do a phase 3 study. And the hypothesis that we base this study on is that ME is a clinical entity that is caused by a malfunctioning of the immune system, probably a kind of or a variant of an autoimmune process, that symptoms can be reversed or improved if the underlying immune defect is treated, that the monoclonal antibody, which is CD20 directed, that is, depletes B lymphocytes, and thus the functions that are connected to this lymphocyte type may result in clinically meaningful responses, at least in subgroups of ME patients. And we saw from the latest phase 2 study that you'll see the details on in +1 in 2 or 3 weeks, that maintenance therapy can give prolonged duration of response compared to induction therapy only, and that we will probably see some patients also responding 24 months into the study, which is the end of the observation period in the planned study. So, when planning a study, you have to have a null hypothesis, and that is that your new treatment is no better than the old one. But what you really are doing, you're trying to confirm a theory or you're trying to refute it. And we are trying to confirm or refute that B lymphocyte depletion with a monoclonal antibody, rituximab, with induction and maintenance treatment, may result in clinically meaningful responses in patients with ME of duration 2 to 15 years. We stopped there because we want to have some kind of homogenization of the patient group. And we didn't want those who possibly could be spontaneously getting better, those who had shorter periods of sickness than 2 years. They had to be fulfilling the Canada criteria. Now, this study is headed and the protocol is made in our institution, but it is a joint effort between 4 university hospitals and a community hospital with special expertise in all the 4 health regions in Norway. And it's recognized and it's accepted both by the Regional Committee for Research Ethics and by the Norwegian Research Council, which is the highest official body founding research in Norway. The patients are locally recruited at each institution, and it's predetermined how many patients that are being recruited at each center, from 40 down to 24, depending on center. All patients are given information beforehand. They are investigated in the center if the interview indicates that they could be eligible. And the patients come to the centers based on interest from themselves or primary health care physicians or specialists or a local hospital. They fill in the consent form for the main study and the sub-studies, and blood tests are taken. It's a major battery, and it takes time to get all the tests in. We want to exclude every other possibility for conditions that may look like ME but not being ME. And the patients, when accepted for the study, they fill in the DePaul questionnaire, but we have put in some additional questions. We are, for example, interested in some subgroups of ME patients. We see that there are some ME patients that seem to have a hyperreactivity in their innate system. They, in fact, never get infections after they get their ME. And we have another group that always get infections. And there's a group in between that's about the same. But, I mean, to us, it seems that this first group is rather large. There's something special with them. We want to know if there's something with this group when we give them treatment that makes them different from others. So we have a lot of clinical information in the files on all patients, and this can be correlated to what we find during the study. We have a biobank, it is sent centrally to Bergen, and it's put down there, and it's under all types of law regulations. The patients fill in symptom severity registration, the fatigue severity scale, and SF-36 at baseline. And then we do the sub-studies for those patients who want to go into sub-studies. The sub-studies differ from institution to institution depending on what we have capacity for doing and what are the local expertises. Patients undergoing ramp exercise tests have to wait at least a few weeks, up to 3 months, to be sure that they're back again in their ordinary condition before getting the first infusion and getting the register of how their condition is. And also it is only a subgroup of patients that are looked upon being fit enough to be included in the study that participate in these ramp exercise tests. And the design of the study, it is randomized. That means it's coincidental what kind of treatment you're given. It's double-blind. That means that you don't know which treatment you're given, and the doctor does not know which treatment is given. And usually in a phase 3 study, you try to compare the new treatment with the established treatment. But when you don't have an established treatment, it is placebo. In this case, it's saline mixed with albumin, so the patient or no one else can see difference when the drug is given. So I feel that we've done all we can to make this a blinded study for everyone involved. The blinding will be kept until the latest included patient has been through the whole observation period for 24 months. And that means in practice that it'll be the end of the summer 2017 when this study can be unblinded, and not before that. The only patient that can be unblinded in the meantime is if we get some very serious side effect, and then we have a safety committee, the safety board of doctors that are not involved in the patients that can go in if it's very imperative to know if the patient has been given an active drug or not, for example, a kind of life-threatening occurrence. Then they can go in that individual patient and find out. Or else the randomization is done externally by a commercial company that is specialized in clinical trials. We have no access to what the patient has been given, and it's done by so-called block randomization. That means that every 8 patients in a single center, we know that within that block, 4 patients have been given Rituximab and 4 patients have been given placebo. But we don't know in which line they come, if it's A, B, 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 A, B, 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 etc. The point of that is that if you come in a situation where a center cannot include all the patients they want, you can stop at a number 8, and then you can send the rest of the patients another place. And you will know that within that same institution, the same number of patients will have Rituximab and will have placebo. It's a way of being sure that you don't get imbalances if there is some type of differences in how you treat patients within the different institutions. It shouldn't be so because the protocol is very strict, but it's a way it's done in many studies. We have external monitoring. It costs money. We have professional people coming from the outside looking at all papers several times during the study to be sure that this is done properly. And once the data is put in the database, the data is locked. We cannot influence the data at all. So that means when the data is there, it's there forever. So we have no impossibility to manipulate the data as we see it. And that is very important, as I will come back to, for the trustworthiness of the results. The schedule given is Rituximab, 500 milligrams per square, day 0 and 14. That is just as in the other studies, but we have gone down in dose, down to 500 milligrams flat, given at 3, 6, 9, and 12 months, and no infusions the second year. But the patients are followed at each infusion. They're seen in beforehand by the doctors, and also they come back to the hospital at 15, 18, 21, and 24 months. The patients themselves do most of the work. They register how the symptoms develop. They look back each 14 days and put up figures for what their symptoms development has been. I won't go into that, but it has worked well in the previous studies. The patients are very good at doing this, so when they understand the system, it functions well. And it tells if there is any change in the fatigue, for example, which is the main endpoint, or the other major disease symptoms that they have. The SF36 is filled in every third month from 0 to 24 months. Fatigue severity scale at 0, 6, 12, and 18 months, 24 months. And we do the electronic registration for 7 consecutive days. That is, the patient has this sensor armband on the upper arm for 7 consecutive days, except when bathing. This is done before start of treatment and 1.5 years into the treatment. It gives a very good estimate of the activity level, and the patients are asked to just do as they normally would do. They're not to do something special. It gives a very good objective indication of the level of activity. You can see in one study we have a patient that had 300 steps per day before study, up to 13,000 steps per day when in response. And that makes more impression on those reading than the patient saying, I'm much more active. But what we see is that what the patients say and what we can read from the electronic devices, they're in very good, they fit very good to each other. Blood banking is done several times at regular intervals during the study. The primary endpoints in the study is the development of the ME fatigue during the 24 months observation time, and the number of patients having clinical responses according to predefined criteria during the treatment. I won't go into details but that's very thoroughly put in the protocol. The secondary endpoints are changes in quality of life, measured by SF36. And when we talk about SF36 in that study that you will see in 2 to 3 weeks, you will see that the major responses, the alterations in SF36 are quite impressive in those patients. In fact, you find SF36 levels at the population normal, in fact, in major responders. So it's something totally different from what we heard about in some other studies that were referred this morning with other methods. So I think that SF36 can be a good measure in these patients. The fatigue severity score at 0 and 1 point into the study, changes in physical performance measured by the armbands at 0 and 18 months, and subjective physical functional level at different points into the study. That is, the patients are given a form which says something about level and what they can manage to do, and they are to estimate how things develop. For example, in that phase 2 study, the patients had around 12 to 15% mean physical function when they went into the study, and good responders often can go up to between 70 and 100% of their pre-ME physical level. Also, the length of response duration and the number of patients still in clinical response at 24 months, and of course also toxicity are secondary endpoints. The sub-studies, as I said, endothelial dysfunction, ability to respond with dilatation of the vessels to adequate stimuli. For large vessels, it's something called FMD, which is flow-mediated vasodilation. It's measured by ultrasound on the brachial artery. When you compress the lower part of the arm for 4 or 5 minutes, open up, and then you measure the dilatation of the brachial artery for the first minute afterwards. Ordinarily, the stimulus will tell that the artery has to send out much more blood. It dilatates. A study from Dundee a few years back showed that these patients with ME, as a group, have endothelial dysfunction, which means that they do not have the ability to dilatate the artery as they should. So we want to do this prospectively, and I'll come a bit back to that. And we also have a system for microvessel investigation in the arm, which is done only in Bergen, while the FMD is done both in Bergen and Notodden. And we aim to see that in a large cohort of patients with well-characterized ME, if there is any evidence of endothelial dysfunction, as we have found in our preliminary studies previously, and as the group in Dundee have found. And if so, is there any correlation between disease severity and FMD? And is there any indication that improvement of the endothelial dysfunction happens in patients that do have clinical responses? I can't say that just looking at the basal data that we have in the patients we have up to now, we can quite certainly say that these patients as a group have endothelial dysfunction. And it also does seem that there is a correlation between severity of disease in the patients as a group and the degree of endothelial dysfunction. But that doesn't mean it is reversed when they have response. That is something we'll have to see. Ergospirometry will be done for 3 places. Patients that are judged to be able to tolerate the bicycle ramp exertion, and that is mainly mild and mild to moderate patients, will be tested for 2 consecutive days before and after, about 18 months into the study. And we are interested to see in a blinded setting if there are changes in anaerobic threshold and other parameters on day 2. Relation between alterations in test results and changes in patient symptoms about 18 months into the study. Does that happen? Is there any correlation between clinical response and, should we say, bettering of these measures, which we know are not functional in this group of patients as a group? I know there are differences between single patients, but as a group, there is something wrong with this system, and that we believe maybe have something to do with the lack of ability to have the autoregulation of the blood flow as it should. We also in Bergen have a sub-study for gastrointestinal function. Our gastroenterologists have become quite interested, and we have a sub-study for those patients that have a lot of gastrointestinal symptoms. I won't go into that. Now the study is underway. It started September 1st last year. We had a good meeting in beforehand with the cooperating 4 other centers. And up to now, 2 of the centers already have finished their recruitment. 2 are doing well, and 1 center started a bit later. It had to do with recruitment of personnel, and they had some logistical problems, but probably all centers will have started infusion by the end of this summer. And that means that summer 2017, we probably will be opening the code and starting to analyze the data. So totally, by the end of May 2015, about 125 patients have been recruited of the 152 that we were to have into the study. And we've had a good dialogue between the different centers and us leaders in Bergen, and decided on problems that have come up, for example, in blood tests and other things, if that stops recruitment for single patients. The cooperation between the pharmacies at the hospital and the researchers is also going well. As you know, The pharmacy has to, of course, know what the patient is to be given, but they get their information from the commercial company and not from us. So that has to function, and up to now it's gone very well. What do we expect to find? I will say that our most important task is to do a study with a high quality. That is more important than getting a special answer. We want to have the true answer. And the quality of the study must be so robust that it can tolerate all types of official external review. FDA, whatever, who want to come, they can come and look at the books and look at all the data and see that this has been done properly. And also, this is a prerequisite if the medical community is to trust the results obtained. Either they come in the one answer or the other. We believe, based on our previous studies, that there will be a subgroup of patients that will have response to rituximab. However, if this can be so many patients that we get a statistically significant positive result of the study, that we don't know. I think that the number of spontaneous improvers in the placebo group during the 24 months will be the key to that. Our impression is that we have very few placebo effects for patients awaiting to become better. For example, we haven't seen a single patient in any of our studies that has had a response starting before 2 months, which you would have expected if it was a placebo effect, which was the important thing. And from oncology and all other areas of medicine, we know that when we do phase 3 studies, we always get poorer results than in phase 2 studies. For example, there can be some bias in inclusion of patients that we haven't thought of, and that is a common experience. So in our study, we have expected, we said 50% of the patients get a clinical response, and 20% of placebo patients have improvement that we can call response according to the criteria. We, of course, when we put up the response, we do not know if the patient has been in the one or the other arm. That is done before the unblinding. So I think probably we will get a lower response rate than the 64, 67% we haven't had in previous studies. That would be not unexpected from previous experience. But we hope maybe that the placebo arm will have a lower response rate than 20%. That is my intuition. If we are so lucky that the study is positive, we can find out a number of other things. Are there clinical characteristics for responders and non-responders that make that, for example, we can say that this patient has a small chance of response to rituximab. We probably won't put him on that drug. That, of course, will spare the patient for unnecessary toxicity, and it will give a higher response rates to those patients who are given the treatment. And is there something characteristic for patients that improve that are placebo patients? Is there something that can characterize those patients? So there's something to learn from that. We also are curious to see if we see what we've seen before, that there's a lag time before response is seen, 4 or 5 months, indicating a kind of washout effect after B-cell depletion, which, as you know, makes us think that autoantibodies may be a part of the mechanism behind the B-cell depletion effect. And we are very glad for having a biobank, because this enables us to work on mechanistic issues in the laboratory for many years. It gives us a possibility to share both information and data and samples with co-investigators within the country and also abroad. We have talked during this meeting about our ongoing cooperation with University College in London and Dr. Scheibenbogen's institution in Berlin, among others. We recently have also got money for a cooperation with the Department of Immunology at the National Hospital in Oslo. I've talked about endothelial dysfunction. It certainly is there, but we don't know if there's a relation between endothelial dysfunction and the symptoms in the patient, or if it's just a kind of thing that characterizes the patients. They also have that. We would, in many ways, expect the endothelial dysfunction to improve when the patients get better, but we don't know that. And what about the ergospirometry? We would expect also there improvement on day 2, but we don't know. No one in the world has done that up till now. And does this study have any political impact? Well, in Norway it has had already, because we have managed to get together a study from 5 different hospitals, including 4 university hospitals, to answer questions concerning ME that we believe are important. And that is quite unique. We have managed to get public financing. The study costs around £2.5 million pounds. That's a lot of money to raise. And we've got money in competition with a lot of other good studies that didn't get money. There, I must say, Øystein Fluge has done a very impressive job working on the protocols and the applications for this. And we got also private financing through the Me and You crowdfunding and the Norwegian ME Association, which we are very grateful for. And I must say that the Kavli Foundation is our lifeline. It's a charitable foundation that came to us in 2011 and said that they wanted to have a cooperation with us, and they have enabled to Øystein to buy him free of my oncology department 2 days a week, and for that he works 4 nights a week and weekends, so he's quite cheap. And we have got a lot of public interest, which has been important for how ME is looked upon in our country. And very many other patients say that. So the 2011 study was very important in that aspect. And I've been invited to the Directorate of Health, for example, and had lectures for them. And that wouldn't have happened a few years ago. And what about internationally? I think it depends on the results. Even with a positive result in the main study, there will usually be a demand for another multi-center study done somewhere else in the world before this is generally accepted as a treatment for ME. And I think it will lead to discussions in many countries if we get a positive study. In Norway, I have the hope that they will not wait for another study somewhere else, because they have put so much money into this. There is so much prestige in it that if this study is well run and it's published in a good publication and it shows a very good result, I think that any patients in Norway will get the opportunity to get Rituximab in the future. If we don't spoil it all with the next study I'm going to tell you about. But, of course, you have to look at toxicity of Rituximab in the long and the short term. What we saw in the phase 2 study was that many patients were still responding after 36 months. But we have 3 patients more that have got gradual recurrence after 36 months. That means that maybe a number of the patients have to have some kind of refill of Rituximab also in the years to come to be kept in response. And of course, keeping the B cells low for years and years, well, we do it in lymphoma, but for a so-called benign condition, that has to be considered. And also we have to look at cost-benefit and estimates of health economy. They will also always ask how much does this improvement of life cost? Can we afford it? Even if we get a primary endpoint that is negative, I think that a well-run cooperative study may pave the way for future intervention studies in ME. I think that if the public view on ME is to change, it's very important for us to try to understand more of the mechanisms behind the disease. So our laboratory studies are very important there. And what we, Øystein and I, have greatest problems with is trying to figure out what are the primary events and what are the secondary events that are going on. Because you've heard a lot of information here today and at the researcher meetings the 2 previous days. A lot of things happen in ME, but what really is the primary event? What is secondary? Where is the initial things happening? I mean, we can use years and years if we don't have a very dedicated look and a well-qualified consideration of what to look at. So we're doing a lot of other studies that haven't been published yet. There will be interesting studies coming out in the years to come. I'm not going into that. But in the meantime, we do have a patient group that does not respond to rituximab. Both the 2 studies, about a third of the patients do not respond. And we have patients that cannot come for rituximab treatment. They get too ill by coming. They cannot tolerate being taken out of the sick home or from their house. Is there a possibility for some other drug? And we have recently started a new study using a very old drug known in oncology for years and years, also used in autoimmune diseases, cyclophosphamide. The reason for interest in this is patients telling about major improvement of ME symptoms during cancer chemotherapy for breast cancer. And looking through the 3 drug combinations that the patients have had, we have guessed that cyclophosphamide is the culprit or maybe the good guy. And we have done a small pilot study in 3 patients, and 2 of the 3 patients had a good response to cyclophosphamide, which works in a different way than rituximab. It works both on the B cells and the T cells. It's an inexpensive drug. Rituximab is very expensive. And of course, if this works nearly as good as rituximab, it will be the preferred drug probably, especially in places in the world that have poor economy. It could be a very good alternative. So we have a 40-patient study, which is just started, where we give pulse cyclophosphamide treatment, 600 milligrams per square, 700 milligrams from the second infusion, 4-week intervals, 6 infusions in all. And it's a single institution study with a small arm at the Norwegian Radium Hospital in Oslo. Our oncology colleagues are getting interested in ME. It's quite interesting. So we have 3 groups of patients. The biggest group is those who have not been treated by drugs in our studies before, 25 of the patients at least. We have the group that has not had response to rituximab. Is it possible that they can respond to another way of manipulating the immune system? And the 3rd group is patients that have had a good response but are deteriorating after some time after rituximab. We do not have any money for treating them again with rituximab at the time being, but they can be candidates for this trial if they want to. So we started in March 2015. We call it popularly cyclome. It's Canadian criteria, at least mild to moderate disease, more than 2 years duration, but they can have been sick for decades. There are some groups we don't want to treat. We follow up for 12 months. It's an open study. Everyone gets cyclophosphamide. The primary endpoints are like in the Rituximab study. And we have the sub-studies with FMV, ergospirometry, like in Rituximab. One of the reasons for this study is we are trying to find something for the sickest patients, those who cannot come for treatment. Cyclophosphamide is possible to give at an advanced primary care facility or in a home for the very sick patients. And we have contact with some dedicated primary care physicians in our country and some smaller hospitals that will cooperate in a study if we get a good response rate in the first study. 20 patients of these very severely ill patients can be given a possibility to participate in this study. But we don't yet know if that will be a study. It's approved, but we have to look at the results in the first study first. And finally, I would like to acknowledge the financial support. It's a joint venture between the Norwegian Research Council, which we applied for money in competition with others. The Department of Health, in fact, gave us money without us asking for it. That's a quite unusual situation. I never had that before. It was just after the 2011 publication, then the Minister of Health came to our department. In fact, over the table said that you're going to have money for this. It's not very usually seen. Regional health authorities, that's also through the Norwegian Research Council and competition there. There were 70 applications for multi-regional studies, and we were one of the 10 selected. So we're also proud of that. The Me&You crowdfunding and the Norwegian ME Association have been very good supporters and helped us. The Kavli Foundation have helped us, especially with the laboratory side of this study. The money for doing all the mechanistic studies will come from them. And we've also been given money by private sponsors. And of course, we also have to say that we have conflicts of interest. We live in a commercialized world, and surveillance over us is something called the BTO, which have decided to make a patent, and the patent's pending on the concept of B lymphocyte depletion in ME. And they've also filed a patent on the use of cyclophosphamide in ME. It's not easy for us to understand how it's possible to make money of that. But as they have done it, we just have to say that is a problem, because Øystein and I are named as inventors. We do not believe that we're going to be rich from these patents. Thank you so much. I'd particularly like to say thank you to InvestMe for inviting us and also for this very nice meeting. I would say we've been here for 5 years, Øystein and I, and this is the best meeting to now. The quality of the meeting has improved. I'm especially impressed by the researcher meeting, which is getting better and better for each year. So thank you very much for that. And also thank you to all our patients, those who are here and those who aren't here, because most of our ideas are in fact derived from listening to patients. It's a good thing for doctors to listen to patients, because what they tell us is the truth. It's our problem to try to derive what is underlying what they tell us, what mechanisms are underlying what they tell us. Because what they tell us is in fact what is happening with them, and that has a cause. So it's up to us to find out what that was. Thank you.
Thank you all. Well, it's very hard to follow such a colossal presentation as that, which gives us so much hope, and how much we admire the skill and intent of your studies. You know, how carefully it's being done is a lesson to everybody who works in these fields. And we wish you well, and we'll be keeping in touch, and we'll be coming to see you. And I know there's going to be a lot of interaction with other scientists. It's very hard to talk about what's happened at the meeting today in the last 2 days. But if anybody has something that they think we might be missing, please say so when you ask Olav questions as well. Because, as Olav has pointed out, it has been quite dramatic for some of us in the last 3 days to watch the enthusiasm, the determination, the good science, the bits of things that are working, things that might work, and so on. That's what science is all about. And I am so impressed by the people who are entering this field, not just the young people who are coming along, but the people who are older and who have been working on this for years. I really do admire them for sticking with it through thick and thin. It's been very hard. When it's easier to walk away from something than stick with it, you know, and you get results like this, we get them to come through. It's so it's so amazing. And I really take my hat off to all the scientists we've heard in the last few days. Keep going, and we will help in every way we can as patients and as just followers of the cause, because I admire all of you, how you've stuck to it when the easiest thing was to walk away and say, well, we're never going to get to the bottom of this. The hell with it, we are. Thank you very much, Olav. So take the question. Olav, you want to... Are there any questions for Olav you want to make, or anybody want to make a point about what they've heard today? Yes, the woman at the back and the gentleman here. Yes, briefly, please. Because I know people need to get home, get their trains and buses and tubes and walk. Hi. Or however. Yeah.
Firstly, just very briefly to say thank you so much. You don't know how grateful we are for your work. My question was, I read somewhere on the internet that at least one of your patients in this trial, you worked out from their response to rituximab that they had a chronic Epstein-Barr virus rather than typical chronic fatigue syndrome, ME. What made you decide that they had chronic... How did you know that they had chronic Epstein-Barr virus?
No, I'm not aware of that.
Okay.
Yeah. That's your answer, right? Okay.
There is some blogging going on, but we're not aware of any such patient in the rituximab study.
Okay. Talk to him afterwards, and you can refer to where you saw it. Yeah. Okay. Gentleman here, please.
You talked about using cyclophosphamide as an alternative to rituximab.
Yes.
Are you yet seeing the same delaying response with cyclophosphamide?
Our experience is rather limited. What we see regularly is that the responders come much faster. We have responses before 3-4 weeks. So the mechanism is a different one, quite certainly.
And are you yet thinking about what that mechanism is?
Well, you know that cyclophosphamide is a broader working drug because with rituximab you get the B-cell depletion, while you have both the B and T cell effects of cyclophosphamide. So it works in a different manner, but we haven't gone closely into that. We have to observe the patients. I mean, our patient experience is very small. We have 3 patients treated with mammary carcinoma. They have all had early responses. The first response was seen the day after the first drug infusion, and the 2 others about 3-4 weeks after being given the drugs. We have patients that have response about 4 weeks, and patients starting to respond around 6 weeks, but which started now in March. So we just have few patients that have started, so we don't have enough information at the time being.
And you seem to indicate that cyclophosphamide is a lot cheaper.
Well, it is, but we don't know if it's as good. We have to see.
We may have to buy it up and make it really cheap, you know.
Well, it is cheap.
If it gets too good, somebody will make money out of it. Not them, but somebody will try to make money. Sir, you have a question.
Yeah. I want to thank you for your presentation. I have one question, because in the first 2 trials, you didn't have a look at B-cell subsets at baseline. Will you do that in the multicentrals trial now?
We will do it, but Øystein, can you say something about that? Well, what Øystein said is that the problem there is that we cannot go into that data. We can take the samples, but we can't look at the analysis, because that will tell us what kind of treatment the patient has been given. So, for example, during this study, we don't do sub-analysis of white blood cells during the whole study, because then that can tell us if they're B-cell depleted or not. So it's not...
Olav, the immunophenotyping data that we examined, we tried to understand after the maintenance study that will be published now. We tried to take new blood samples after 40 months to see is there any difference in B-cell regeneration, the subsets for those responding, still responding, and those with relapse. And the same panel that we analyzed and had some preliminary data, that will be done at baseline and at 18, 21, and 24 months in the randomized study, so that when the B cells are starting to regenerate, we can see is there any difference between those relapsing early versus those with sustained response.
Okay. Thank you.